Well, thanks very much, Jeremy. Thank you all for having me. This is in terms of the lineage I'm going to be talking about, it's a bit of a shift from most of the talks so far, and we're going over into the plant kingdom. But given the very close correspondence or relationships between insects in particular and flowering plants, both in an ecological and an evolutionary sense, I hope some of the audience find something of interest in this talk. So I'm speaking today from Ku-ring-gai country in the northern Sydney basin, and I acknowledge the traditional owners of this country, past, present and emerging. So I'm going to be talking about a large project, the Australian Angiosperm Tree of Life project that's been running for a couple of years now, and just report on where we've gotten to so far and where we hope to go to in the near future. It's been a big project with a lot of stakeholders, so this talk has a very large authorship, and I'm not going to go through every one of those names, but there are all their affiliations as well. So the work I'm reporting on today, as I said, is part of a much larger project, which was launched around 2 years ago at the Brisbane ASBS conference, and that's the Genomics for Australian Plants initiative. This whole project was really catalysed by Bioplatforms Australia, and in partnership with most, if not all, of the major herbaria and botanic gardens around the country. The aim of the broad project is to develop genomics resources to help us as practitioners and scientists understand the evolution and the conservation of our flora. Within the broad GAP umbrella, there are three main streams of activity. There's a reference genome stream, a conservation genomics stream, and a phylogenomics stream, which is what I'll be talking about today. So to conceive and drive the phylogenomics project, or the phylogenomics stream of GAP, we assembled a working group a couple of years ago, and here are all the members of that working group. And the working group conceived an ambitious project. And the aim over the long term of this is to resolve the Australian Angiosperm Tree of Life to species level. And a species level tree would be something in the order of 23,000 tips. So it's an ambitious goal, and we resolved to work towards it in 3 stages. The first stage is to resolve the tree to the genus level, complete for at least 95% of accepted native genera. The second stage was to dive more deeply into clades of interest and sample much more densely to address questions that we always ask in our field, and that is, are these groups monophyletic? What's their evolution? What's their biogeography? Does taxonomy reflect monophyly and all the rest of it? And then the third stage is to then fill out all of those unsampled tips and resolve the tree to species level, to a 95% completeness level. So today I'm just talking about progress against stage 1. So at the outset, we established some key principles for this broad project. Because it involves the collections community, we were adamant that whatever we do has to add value to our collections. We also insisted on gold standard sample documentation. So every sample submitted has to be vouched by a specimen that is already accessioned into a collection and not sitting in someone's office. We insisted on detailed metadata and that the samples, in terms of how they were collected, are compliant with the Nagoya Protocol. And that the names under which samples were submitted were compliant with the Australian Plant Census. There was a fairly substantial requirement for researcher co-investment with this project, so it's not a grant scheme. And as it turned out, approximately 2/3 or up to 2/3 of the dollar cost of generating data for a single sample was actually borne by the participants, by the community itself. So it was fairly substantial investment by the participants. We insisted on open data principles, so all the raw data will be available after a 12-month embargo publicly. And overall, we wanted to really build a platform for the future. So not only did we want to do some good science, but we also wanted to generate data that are the highest possible quality and are maximally reusable. And also to, through this process, upskill our community in not only the generation and interpretation of phylogenomic data, but also their use. Okay, so the implementation of this stage 1, which is the genus level tree, is still a fairly substantial undertaking. So we approached it from a community consortium perspective. So the participants across all of the botanic gardens and the herbaria organised themselves essentially into 6 teams, and those 6 teams were centred on the 6 largest Australian herbaria, and were led by these 6 individuals. But of course, many other people were involved. So here you've got pictured some of the collections management staff, other researchers, PhD students, bioinformaticians, and project managers. And very early on in the conception of this project, we decided to partner with an existing project that is similar in design, but global in scope. And that project is the Plant and Fungal Trees of Life project, being coordinated by the Royal Botanic Gardens in Kew in London. So partnering with PAFTOL ensured that we could deliver a much more complete sampling of Australian taxa than PAFTOL proposed to achieve alone. They were only going to get to about 50% representation of the Australian flora. Partnering with them also meant we could share resources to maximise sampling efficiency and progress. And it also ensured that we had access to samples of all of the non-Australian lineages that we were not able to sample because our project is funded by platforms through NCRIS, which means that we can only sequence Australian samples. So partnering with PAFTOL meant that we got access to all of those out-group and in-group lineages that are non-Australian, which of course is critical for any question that relates to monophyly. And we took a target sequence capture approach to build our big genomic data set, our phylogenomic data set. And we were fortunate that not long ago, a really, really useful universal probe set is available for angiosperms that targets 353 low copy nuclear genes. And that kit's commercially available. So that's the structure of the project. What have we done so far? Well, COVID lockdowns, I guess it's moot to say, have played havoc with our timelines, but we've still managed to make, I think, really great progress. So currently, we've managed to sequence nearly 1,400 Australian genera through the GAP project. And those raw sequence data are already available on the GAP portal, available to consortium members at least, and will be made broadly publicly available after a 12-month embargo. Importantly, of those 1,400 odd samples, about 300 were not represented by any sequences in public databases. So as far as we know, they've never been sequenced before. PAFTOL, as part of our partnership, contributed another 545 genera to our combined data set. So altogether, we got to about 93% of the over 2,000 genera of Australian angiosperms, flowering plants, which I think is, for a first pass, a pretty good result. Geographically, all of our samples cover the continent, so we didn't have a narrow concentration on areas around capital cities, for example. And here's a histogram summarising the gene recovery of our raw sequence data for those nearly 2,000 samples that GAP sequenced. Now, we could recover a maximum of 353 genes because that's what the baits were targeting. And you can see that for over 2/3 of our samples, more than 200 of those 350 genes were recovered. Only 9% of our samples yielded fewer than 50 genes. Now, 50 genes is still a lot of nuclear data, much more than we've traditionally generated with Sanger approaches. And then, of course, there's always a group of samples that just don't work for whatever reason. And so there are about 75, nearly 80 samples for which we got fewer than 10 genes. And these represent the failures, I guess. But given that the bulk of our samples, the overwhelming majority of our samples were sourced from herbarium specimens, that's a really satisfyingly low failure rate. And of course, gene recovery varied from lineage to lineage. So the orchids, for example, yielded fairly low relative gene recovery, so mostly below about 150 genes per sample, whereas the grasses, the brassicas, and the mints, the lamiaceae, all showed very high relative gene recovery, mostly over about 300 genes per sample. So that's the raw data. Well, of course, what everyone wants to know is, are they any good for resolving phylogenies? And I'm happy to report that it appears to be a big yes. So this is our very first very rough tree. We've pulled this one together with just a very fast tree search algorithm, FastTree. And we haven't really done, or we're in the process now of doing the rigorous sort of sample interrogation process to determine which samples should be removed for the next round of analysis. So this is a very rough and dirty tree. All caveats aside, there's an awful lot to like about this. So I've indicated on the tree there where the major Australian families are positioned. And I can say with some confidence, the great majority of samples go in the right families, and the families go in the right positions relative to each other based on what we know about angiosperm phylogeny built up over the last 20-odd years. So what else have we done apart from the tree? Well, as I say, a lot of this project was to do with not only building science outputs, but upskilling the community. So some other activities that have been going on. Chris Jackson at the Royal Botanic Gardens in Melbourne has developed a new data processing pipeline, which is basically HivePiper, which anyone who works with phylogenomic data, target capture data, will probably have heard of. So integrating a paralogy resolution method with the HivePiper pipeline to build this consolidated pipeline. The paper describing the pipeline's up on bioRxiv now. And of course, paralogy is a real problem with plant genomes to a far greater extent than most animal genomes, because whole genome duplication and hybridization have been very powerful and pervasive forces in the evolution of plants. We've also run some workshops associated with the ASBS conference earlier this year, some webinars that are available on the GAP website on how to understand and treat multi-gene data sets and phasing hybrid accessions. But most importantly, I think this project has successfully, I suppose, prosecuted a really broad and deep collaboration. So we've got over 70 participants from across the Australian institutions, botanical institutions, all government herbaria are involved, 4 university collections are involved. And it's just been really great to work with all of these wonderful people towards a common goal. What are our next steps? As I mentioned before, we're going to further interrogate questionable samples, resolve paralogy to the extent that we can. We're also aiming to recover plastid genes. These, of course, were not the targets of the bait kit, but they're in such abundance in plant cells that you can often pull them out anyway. Then phylogenetic analyses, writing papers, and so on and so forth. Stage 2 of GAP, the GAP phylogenomics project, the ATOL project, which is the one where we get down into the detail of specific clades of interest, is now underway. So we've got 16 projects, community-based projects on specific clades underway there. And stage 3 hasn't yet begun. I can see I'm running out of time, so coming up to the end now. So just to summarise progress so far, stage 1, complete for data generation at least, but still some analyses to do. Stage 2, the clade level studies underway, 16 projects there ongoing. And stage 3 at this stage is unfunded and effectively a blue sky idea. But we're going to be working very hard over the next year or two to pull together what we need to do to make an attractive bid for a fairly substantial amount of funding that'll be required to complete that. We'd have to do about 20,000 samples, around 200 plates worth. But we figure the science outputs for a species level phylogeny of the Australian flora are absolutely worth the work. So lots of people to thank, and here are just some of them. Thanks very much. I haven't left much time for questions, but I'm happy to answer any in the chat.